Paper O3 Biodiversity and Conservation. 1. Introduction. Biodiversity constitutes the most important working component of a natural ecosystem. It helps to maintain ecological processes and deals with the degree of nature's variety in the biosphere. The term biodiversity was first coined by Water and Rosen in 1985. Biodiversity refers to the variability among living organisms and constitute terrestrial, marine, aquatic ecosystems and the ecological complexes. It reflects the number, variety and variability of living organisms and includes diversity within species, genetic diversity between species, species diversity, and between ecosystems, ecosystem diversity. India is a mega-diverse country having only 2.4% of the world's land area but accommodates 7-8% species of flora and fauna. It includes more than 45,000 species of plants and 91,000 species of animals. It is situated at the tri-junction of the Afro-tropical, Indo-Malayan and Palearctic realms, all of these support rich biodiversity. India is a diverse nation, which has 10 biogeographic zones. A comparison of status of biodiversity between Indian and world scenario is depicted in Table 1. Table 1. Comparison between the number of species in India and the world group number of species in India. Number of species in the world. Percent India. Mammals 354 6277.6. Birds 1224 9702 12.6 Reptiles 408 6550 6.2 Amphibians 197 4522 4.4 Fishes 2546 21730 11.7 Flowering plants 15002 50006 Source: Center for Ecological Sciences, Indian Institute of Science. India is also a recognized center of crop diversity and produces hundreds of varieties of crop plants such as rice, maize, millets, etc. The diverse physical features and climatic conditions of India are responsible for a variety of ecosystems such as forests, grasslands, wetlands, deserts, coastal and marine ecosystems. Paper O3 Biodiversity and Conservation. 2. Biodiversity hotspots. The idea of hotspots was first given by ecologist Norman Myers in 1988. Biodiversity hotspot is a biogeographic region that contains significant reservoir of biodiversity and is under threat and destruction. The main criteria for determining a hotspot are endemism, the presence of species found nowhere else on Earth, and degree of threat. India has 4 hotspots out of 34 identified globally, Conservation International 2013, which are follows. Himalaya, comprises the entire Indian Himalayan region and that falling in Pakistan, Tibet, Nepal, Bhutan, China and Myanmar. B. Indo-Burma, contains entire northeastern India, except Assam and Andaman group. Pakistan, Tibet, Nepal, Bhutan, China and Myanmar. B. Indo-Burma, contains entire northeastern India, except Assam and Andaman group. Of islands and Myanmar, Thailand, Vietnam, Laos, Cambodia and southern China. See Sundalands, includes Nicobar group of islands and Indonesia, Malaysia, Singapore. Brunei, Philippines. D. Western Ghats and Sri Lanka, includes entire Western Ghats and Sri Lanka. The International Union for Conservation of Nature (IUCN) is a leading international organization working in the field of nature conservation and sustainable use of natural resources. It was established in 1948 and evolved as world's largest environment network and plays a key role to save biodiversity at global level. IUCN includes over 1,400 members of both governmental and non-governmental organizations. The IUCN publishes IUCN Red List of Threatened Species. In 1969, India became a state member of IUCN. The office of IUCN India located in New Delhi was established in 2007. The IUCN Red List of Threatened Species, founded in 1964, is the world's most comprehensive inventory of the global conservation status of biological species. As per the relative risk of extinction, the Red Data List contains 7 categories. There are categories given below along with some Indian species. Extinct X, no known individuals remaining. Extinct in the wild EU, known only to survive in captivity, or as a naturalized population outside its historic range. Critically endangered CR, extremely high risk of extinction in the wild, endangered, and high risk of extinction in the wild. Vulnerable VU, high risk of endangerment in the wild, near threatened NT, likely to become endangered in the near future, least concern LC, or lowest risk does not qualify for a more at risk category. Paper O3 Biodiversity and Conservation. 3. Animal biodiversity. India has over 6.7% of the total animal species of the world. This includes approximately 96,373 known animal species and 63,423 insect species. The number of threatened fauna species in different categories and those listed in the International Union for Conservation of Nature IUCN 2014-15 are given in Figure 1 and 2. Figure 1. Representation of evaluated Indian fauna N equals 4681 under IUCN 2014-15 threat categories. Source: India's 5th National Report to the Convention on Biological Diversity, MOF, GOI. Figure 2. Indian threatened fauna. Source: India's 5th National Report to the Convention on Biological Diversity, MOF, GOI. 0500 1000 1500 2000 2500 3000 3500. Least concern. Lower risk vulnerable. Near threatened. Endangered critically endangered extinct. Number of species. 050 100 150 200 250. Birds. Reptiles amphibians. Fishes mollusks mammals other. Number of species. N. MA. E. FO. S. EP. IC. SE. Paper O3 Biodiversity and Conservation. 4. Plant biodiversity. With an estimated 29,105 species of algae, bryophytes, pteridophytes, gymnosperms and angiosperms, India holds 9.13% of the world's known floral diversity in these groups. The richness of Indian plant species as compared with the world is shown in Table 2 and risks categories are depicted in Figure 3. Table 2. Indian plant species as compared with the world. India world % India. Algae 7244-40800, 17.75. Bryophytes 2504-14500, 17.27. Pteridophytes 1267-12000, 10.56. Gymnosperms 74,650, 11.38. Angiosperms 17926-25000, 7.17. Total 29015-317950, 9.13. Source: India's 5th National Report to the Convention on Biological Diversity, MOF, GOI. Figure 3. Representation of evaluated Indian flora N equals 1218 under IUCN 2014-15 threat categories. Source: India's 5th National Report to the Convention on Biological Diversity, MOF, GOI. 0 100 septillion 200 sextillion 300 quintillion 400 quadrillion 500 trillion 600 billion 700 million 800,000 900. Extinct. Extinct in the wild. Critically endangered. Vulnerable. Near threatened. Lower risk. Least concern. No of species. Paper 03 Biodiversity and Conservation. 5. Marine biodiversity. India's coastal and marine areas hold many biological treasures. Dense mangrove forest of Sundarbans, world's largest congregations of nesting turtles in Odisha, beautiful seagrass beds in Palk Bay, dolphins and dugongs in the Gulf of Mannar, majestic whale sharks in the Gulf of Kachach are just a few examples of the treasures of India's coastal and marine biodiversity. The marine floral and faunal biodiversity of India is remarkable, with over 200 diatom species, 90 dinoflagellates, 844 marine algae, 560 species of corals, 39 mangrove species, more than 10,000 invertebrate species and 2,500 vertebrate species. 6. Endemism species in India. In terms of endemism of vertebrate groups, India's global ranking is 10th in birds with 69 species, 5th in reptiles with 156 species and 7th in amphibians with 110 species. Endemism is manifested most prominently in the Indian fauna among the amphibia, 61.2%, and reptilia, 47%. It is estimated that 62% of the known amphibian species are endemic to India, of which a majority are found in the Western Ghats source. Endemism in plants is significant across different plant groups in India. India accommodates 47 families, 141 genera and about 4,045 species of flowering plants angiosperms. These are distributed in the floristically rich areas of northeast India, the Western Ghats, the Northwest Himalaya and the Andaman and Nicobar Islands. 7. Fish biodiversity. Zoological Survey of India, ZSI, has recorded 3,022 species which constitute about 9.41% of the known fish species of the world. National Bureau of Fish Genetic Resources, NBFGR, is mandated to carry out research related to cataloging, characterization and conservation of country's fish germplasm resources. The Bureau has built up a database of about 2,553 native fish species of India, generated information on population genetic structure of 23 prioritized endemic and commercially important species. 8. Critically endangered species in India. Critically endangered is the highest risk category assigned by the IUCN, International Union for Conservation of Nature, which comes under Red List. Table 3 lists critically endangered species, which include birds, reptiles, mammals, fishes, spiders along with distribution in India. Paper 03 Biodiversity and Conservation. There are 5 quantitative criteria to determine wild species, whether a taxon is under-threatened. A taxon is under-critically endangered if meets any of the following criteria. I. Populations have declined or will decrease, by greater than 80% over the last 10 years are 3 generations. 2. Have a restricted geographical range. 3. Small population size of less than 250 individuals and continuing decline at 25% in 3 years or 1 generation. IV. Very small or restricted population of fewer than 50 mature individuals. V. High probability of extinction in the wild. Table 3. Critically endangered species in India, MOEF Report 2011. Birds. Common name. Zoological name distribution. Jerdon's courser Rhinoptilus bitorquatus, Andhra Pradesh. Forest owlet Heteroglobuity, South Madhya Pradesh, in northwest Maharashtra and north-central Maharashtra. White-bellied heron Ardea insignis, Bhutan and northeast India to the hills of Bangladesh and North Myanmar. White-backed vulture, slender-billed vulture, long-billed vulture, red-headed vulture, Gyps bengalensis, Gyps tenurostrus, Gyps indicus. Sarcogypscalvis, across India. Bengal florican Huberopsis bengalensis, Uttar Pradesh, Assam and Arunachal Pradesh. Himalayan quail Ophrys superciliosa, western Himalayas. Pink-headed duck Rhodonis acariophilacea, northeast India. Sociable lapwing Vanellus gregarious, north and northwest of the country. Spoon-billed sandpiper Urinarynchus pygmaeus, West Bengal, Orissa, Kerala and Tamil Nadu. Siberian crane Grus leucogerinus Kaoladeo National Park in Rajasthan. Mammals. Pygmy hog Perculus albania, southern Himalayan foothills but now restricted to only a single remnant. Population in Manas Wildlife Sanctuary. Andaman white-toothed shrew. Crocodera andamanensis, Andaman Islands. Jenkins Andaman spiny shrew. Crocodera jenkinsi, Andaman Islands. Nicobar white-tailed shrew. Crocodera nicobarica, Nicobar Island. Kondana rat Millardia kondana near Pune in Maharashtra. Paper 03 Biodiversity and Conservation. Large rock rat or Elvira rat. Cremnemes elvira, eastern Ghats of Tamil Nadu. Namdappa Biswamoya Terrace Biswasi Namdapha Tiger Reserve in Arunachal Pradesh Malabar Civit Vibra Civitina Western Ghats Sumatran Rhinoceros Dicerorhinus sumatrensis foothills of the Himalayas in northeast India. Reptiles. Gharial Gabulus gangeticus National Chambal Sanctuary, spread across 3 states of Uttar Pradesh, Rajasthan and Madhya Pradesh in India. Hawksbill turtle Erythmochilus imbricata Andaman and Nicobar Islands, the coast of Tamil Nadu and Orissa leatherback turtle Dermochilus coriacea Atlantic, Pacific, and Indian Oceans River Terrapin Badagu or Baska Indian River. Red-crowned roofed turtle. Badagur Kachuga watershed of the Ganga. Cisperidae gecko Cemaspis cisparensis Western Ghats, and found in Cispera, Nilgiris, Cavali near Cochin. Amphibians. Anomalai flying frog Rhacophorus pseudo-malabaricus. Andipuri Shola, Pudothotum and the Anomalai hills of Tamil Nadu and Kerala. Gundia Indian frog Indirana Gundia Gundia, Kempfuli in the Western Ghats. Region of Karnataka, South India. Kerala Indian frog Indirana Phronoderma Anomalai hills of Kerala and Tamil Nadu in the Western Ghats of South India. Charles Darwin's frog Indirana Charles Darwini Mount Harriet in South Andaman Island and Saddle Peak in the North Andaman Island. Kaditar bubble nest frog. Microxylus kaditarensis Hassan district in Badra in Chikmagalur district, Karnataka, India. Amboli bush frog Pseudophylotis Amboli Maharashtra, Karnataka Chalazodes bubble nest frog. Roarchests Chalazodes Western Ghats. Small bush frog Roarchests Chada Tiruvananthapuram district of Kerala Green-eyed bush frog Roarchests Chlorosama Munnar, Idukki district, Kerala I heat bush frog Roarchests heat Munnar, Devakulam and Bagaman in Idukki district of Kerala, and Anomalai hills and Valparai in Coimbatore district of Tamil Nadu. Kaikats bush frog Roarchests Kaikati Nelliampathy in Palakkad district of Kerala Marks bush frog Roarchests Marki Kaikati Nelliampathy in Palakkad district. Kerala, India Munnar bush frog Roarchests Munnarensis Devakulam in Munnar, Idukki district. Paper 03 Biodiversity and Conservation. Kerala. Sacred Grove bush frog Roarchests Sancti Sylvaticus Kapilthara Falls, Amarkantak, Jabalpur. District, Madhya Pradesh tiger toad Xanthophrine tigrinus Amboli, Sindhudurg district, Maharashtra Shillong bubble nest frog Roarchests Shillongensis Malki forest, Shillong, Meghalaya and in Mizoram. Fish. Pondicherry shark Carcharinus hemiode in Indian Ocean, Hooghly River Ganges shark Glyphus gangeticus the Ganga River system and Hooghly River knife-toothed sawfish and Oxypristis cuspidata widespread in western part of the Indo-Pacific region, including Red Sea large-toothed sawfish Pristis microt in western part of the Indo-Pacific longcomb sawfish or narrow snout sawfish. Pristis Zachron Indo-Pacific region. Spiders. Rameshwaram ornamental. Poecilotheria hanumavilasumica coastal savanna, tropical lowland rain forests and montane forests up to an altitude of 2000 m above mean sea level. Peacock tarantula Poecilotheria metallica endemic to India. Corals. Fire corals Millipora boschmai possibility extinct in India. The IUCN list published in 2015 included 988 species of India in Red Data Book. It was 973 in year 2014 and 659 species in 2008. In Indian context, the IUCN list 2015 classified mammals to critically endangered and endangered species. Critically endangered mammals. Himalayan brown red bear Ursus arctos isabellinus pygmy hog Perculus albania. Andaman white-toothed shrew Crocodera andamanensis Condana rat Millardia condana. Large rock rat or Elvira rat Cremnemes elvira, Namdapha flying squirrel, Biswamoya Terrace Biswasi, Malabar civet Vibra civitina. Sumatran rhinoceros Dicerorhinus sumatrensis Kashmir stag Hongle Cervus alophus hanglu. Paper 03 Biodiversity and Conservation. Endangered, mammals, red panda. Wild ass cur Equus hemionus cur. Dole Asiatic wild dog or Indian wild dog Goon alpinus Elds deer Thomon or brow antlered deer Panalia eldi golden langur Trachypithecus gii. Himalayan white-bellied musk deer Moschus leucogaster, hispid hare Assam rabbit Caprologus hispidus. Hog deer Hyelaphus porcinus. Lion-tailed macaque Wanderoo Macaca silenus. Cheetah Tibetan antelope Panthalops hodgsoni. Nilgiri langur Nilgiri leaf monkey Trachypithecus johnny, Nilgiri tar Nilgiri tragus hylocrius. Endangered, marine mammals, freshwater river dolphin. Ganges river dolphin Platanista gangetica, Indus river dolphin Platanista gangetica minor. 9. Agricultural crops and domesticated animal biodiversity. India is basically an agriculture-based county. It has 15 different agroclimatic zones. India is also considered to be a center of origin of rice. India also has a vast and rich repository of farm animals, represented by a broad spectrum of native breeds of cattle 34 buffaloes 12 goat 21 sheep 39, and chicken 15. To conserve India's rich domesticated biodiversity and promote crops and animal breeds, the Ministry of Agriculture has set up 6 national bureaus namely, Plant Genetic Resources, Animal Genetic Resources, Fish Genetic Resources, Agriculturally Important Insects, Agriculturally Important Microorganisms and Soil Sciences. These bureaus serve as the nodal organizations for characterization, evaluation, cataloging and establishing of national databases of living organisms corresponding to their mandates. Apart from this, India has also preserved billions of diverse microbes, many of which are found nowhere else in the world. The country is also accomplished with enormous variability in agriculturally important microorganisms. Source: India's 5th National Report to the Convention on Biological Diversity, MOF, GOI. Paper 03 Biodiversity and Conservation. 10. Biodiversity conservation in India, protected areas, national parks, sanctuaries, conservation and community reserves. India has a very long tradition of setting areas for conservation of wild flora and fauna. After independence, several protected areas were designated in the form of national parks and wildlife sanctuaries. Protected areas are those in which human interference and the exploitation of resources is restricted. Protected areas are categorized depending upon the level of protection, governed by the laws of each country or the regulations of the international organizations involved. Currently in the form of protected area, India form a network of 103 national parks covering 40,500.13 kilometers and 544 sanctuaries covering 118,931.80 kilometers, 76 conservation reserves and 46 community reserves. Table 4. State-wise national parks in different states of India is depicted in Figure 4. Table 4. Details of protected areas in India. Year. No. Of. National parks. Area under national parks. km. No. Of wildlife. Sanctuaries area under wildlife sanctuaries number. Of. Community Y reserves area under community reserves. No. Of. Conservation reserves area. Under. Conservation. Reserves number. Of. Protected areas total area under. Protected areas. 2018-103-40500.13544-118,931.804672.55669,162,072.49. Source: National Wildlife Database Cell, Wildlife Institute of India. Figure 4. No of national parks in different states of India. Source: National Wildlife Database Cell, Wildlife Institute of India. 5. 3. 4. 5. 4. 5. 6. 9. 6. 2. 5. 5. 5. 6. 6. 9. A. DN. RH. A. UR. AN. HC. A. SA. ASM. IB. HC. TT. SI. AG. R. G. O. G. JU. RA. A. H. RA. AY. N. H. MI. CA. and J. HJ. RA. HK. NA. AC. NR. TA. KA. A. EC. AR. AL. M. DA. YH. A. RP. DA. SEH. M. HA. RA. SA. TH. R. M. NA. PI. U. M. GE. AL. Y. M. CI. Paper 03 Biodiversity and Conservation. 11. Wetlands in India. India is bestowed with a rich diversity of wetlands, ranging from high-altitude lakes of the Himalayas, floodplains and marshes of the Gangetic-Brahmaputra alluvial plains, saline flats of green Indian desert to extensive mangroves marshes bordering the country's east and west coastline. As per remote sensing imagery-based assessment, India has total of 26 Ramsar wetland sites covering a total area of 12,119.03 square kilometers. List of Ramsar sites in India is given in the Table 5. Table 5. List of Ramsar sites in India. No. Name of site, state, location, date of declaration. Area. In SQ.km. 1 is the Moody Wetland, Kerala, the 19th of August 2002, 18602 B Tarkanika Mangroves, Orissa, the 19th of August 2002, 5253 Boj Wetlands, Madhya Pradesh, the 19th of August 2002, 314 Chanderdal Wetland, Himachal Pradesh, the 11th of August 2005, 38.565 Chilka Lake, Orissa, the 10th of January 1981, 11406 Deepurbil, Assam, the 19th of August 2002, 4.147 East Calcutta Wetlands, West Bengal, the 19th of August 2002, 3788 Harike Lake, Punjab, the 23rd of March 1998, 69 Hokara Wetland, Jammu and Kashmir, the 11th of August 2005, 13.75 10 Kanjli Lake, Punjab, the 22nd of January 2002, 14.84 11 Keoladeo, Ghana, NP, Rajasthan, the 10th of January 1981, 28.73 12 Kalaru Lake, Andhra Pradesh, the 19th of August 2002, 673 13 Loktak Lake, Manipur, the 23rd of March 1999, 45 14 Nalsaravar. Bird Sanctuary Gujarat, the 12th of September 24 1 2015. Kalimer Tamil Nadu, the 19th of August 2002, 17.26 16 Palm Dam Lake, Himachal Pradesh, the 19th of August 2002, 307.29 17 Renuka Wetland, Himachal Pradesh, the 11th of August 2005, not available 18 Ropar Lake, Punjab, the 22nd of January 2002, 41.36 19 Rudrasagar Lake, Tripura, the 11th of August 2005, 2.40 20 Sambar Lake, Rajasthan, the 23rd of March 1997 36. Paper 03 Biodiversity and Conservation. 21 Sasthamkota Lake, Kerala, the 19th of August 2002, 11.322 Saransar Mansar Lakes, Jammu and Kashmir, the 11th of August 2005, 3.50 23 Samoriri Lake, Jammu and Kashmir, the 19th of August 2002, 12024 Bembanakal Wetland, Kerala, the 19th of August 2002, 458325 Upper Ganga River. Brai Hat to Narora Stretch. Uttar Pradesh, the 11th of August 2005, 265.90. 26 Walar Lake, Jammu and Kashmir, the 23rd of March 1991, 73. Total area SQ km 12,119.03. Source: Ministry of Environment and Forests and Climate Change, Government of India. Date of access: the 2nd of February 2018. 12. Government Acts. Biological Diversity Act 2002. The Biological Diversity Act 2002 was born in India to resultant of India's attempt to achieve objectives, which determined the United Nations Convention on Biological Diversity, CBD, 1992 for protect own biological resources. The Act aims to conserve biological resources with a sustainable manner. For purposes of implementing the objectives of this Act, established the National Biodiversity Authority in Chennai on 2003. India successfully hosted the 11th meeting of the Conference of the Parties COP11 to the Convention on Biological Diversity, CBD, held from 8 to 19 October 2012, in Hyderabad. 13. Other initiatives. Capacity Building for Industrial Pollution Management, CBIPM. Compensatory Afforestation Fund Management and Planning Authority, CAMPA. National Center for Sustainable Coastal Management, NCSCM. Conservation of Natural Resources and Ecosystems. Environment Information, Education and Awareness. Environmental Management and Heritage, Pilgrimage and Tourist Centers including Taj Protection. Social Forestry with Communities, Panchayat Ban Yojana. Paper O3 Biodiversity and Conservation. 1. Introduction. Biodiversity constitutes the most important working component of a natural ecosystem. It helps to maintain ecological processes and deals with the degree of nature's variety in the biosphere. The term biodiversity was first coined by Water and Rosen in 1985. Biodiversity refers to the variability among living organisms and constitute terrestrial, marine, aquatic ecosystems and the ecological complexes. It reflects the number, variety and variability of living organisms and includes diversity within species, genetic diversity between species, species diversity, and between ecosystems, ecosystem diversity. India is a mega-diverse country having only 2.4% of the world's land area but accommodates 7-8% species of flora and fauna. It includes more than 45,000 species of plants and 91,000 species of animals. It is situated at the tri-junction of the Afro-tropical, Indo-Malayan and Palearctic realms. All of these support rich biodiversity. Five. Paper O3 Biodiversity and Conservation. 3. Description. 3.1. Introduction. Species is the basic and finest unit for both taxonomy and biodiversity. Characterization of biodiversity de facto means characterization of the species composition, its alpha taxonomy. The discipline of taxonomy encompasses characterization, classification and naming of taxa, especially species. At present, world's biodiversity encompasses approximately 2 million named species, but that is only a tip of giant iceberg. It is now considered that more than 90% of species on this planet remain to be discovered and described at length. At present, there is a severe shortage of taxonomists throughout the world, along with declining funds for taxonomic research, a phenomenon known among taxonomists as taxonomic impediment. Taxonomy had traditionally been regarded as a dry subject. As more and more scientists started believing that the scientific research should be utility-driven, asking for the usefulness of a particular research for the betterment of humanity, and commercial values, the discipline of taxonomy lost its glory in contrast with high-impact research fields like molecular biology. The implications were profound. The field in which this negative trend had affected the most was arguably biodiversity characterization. There exists an imminent risk that most of the undescribed biodiversity might go extinct even before we get a chance to describe it. In addition, there are several biases at play with respect to the biodiversity characterization. Most of the taxonomists are based in richer countries of temperate northern hemisphere, comparatively less speciose biogeographical zone. Most of the biodiversity on planet Earth is at the tropics. However, tropical countries neither have resources to support extensive taxonomic surveys, nor trained taxonomists. Because of this, most of the described taxa in the world are from that of the rich countries, and is obviously a skewed representation of the real biodiversity. Yet another bias is that most of the described biodiversity is that of the terrestrial biomes, albeit rich in biodiversity and more than 70% of Earth's surface being ocean, only a tiny fraction. 6. Paper 03 Biodiversity and Conservation. Of the marine biodiversity had been described till date. Yet another disturbing pattern in the described biodiversity is that while certain taxa like angiosperms are very well represented, taxa like prasinophytes or terrestrial algae are extremely underrepresented. This is because there are several experts in the field of angiosperm taxonomy, while very few taxonomists working on prasinophytes or terrestrial algae, although the biodiversity of all these three lineages are expected to be similar. These underrepresented lineages and underrepresented habitats of the world need to be prioritized for biodiversity characterization to be effective. The terms biodiversity characterization and inventorying are used interchangeably to refer documentation of biological diversity at the species level. However, there is a subtle difference between the two. Characterization refers documentation of the taxonomic information, for example, species discovery report of the new species, along with its naming, nomenclature discovery of cryptic species discovery that one species is a complex of two species or taxonomic synonymy, that two taxa, for example, two species are actually one, and so on. Biodiversity characterization essentially is part of the discipline taxonomy. On the other hand, biodiversity inventorying refers to documenting overall species diversity of a particular area, for example 1 meter square quadrat at a tropical rainforest, or at a seaweed bed. Inventorying generates a list of known species present at a location. No new taxonomically relevant information such as species discovery, discovery of cryptic species or species synonymy is generated. However, inventorying can generate new records, identification of known species at a new location where it had never been reported. For example, documentation of an invasive species at a new location where it had never been reported previously. Biodiversity inventories generate the so-called checklists, a simple list of all species at a given location. While biodiversity characterization is far more complicated in terms of taxonomy, generation of checklists requires more rigorous fieldwork, and both are equally valuable data regarding biodiversity documentation is concerned. 7. Paper 03 Biodiversity and Conservation. 3.2. Species inventorying. The cornerstone of species inventorying is the generation of species checklists of a particular location. Perhaps the most valuable among different forms of checklists is all taxa biodiversity inventory (ATBI), documentation of each species at a given location including animals, plants, eukaryotes, microbes, prokaryotes, viruses, prions, etc. Generation of ATBI is therefore a daunting task, and always incomplete. This is because bacterial, viral and prion diversity at a given area as little as a centimeter square would be in billions. Most of the microorganisms cannot be cultured in artificial media, and therefore to document its hidden diversity, newer culture-independent methods like environmental DNA metagenomics need to be incorporated. Far simpler approach for species inventorying is to target a specific taxonomic hierarchy, for example checklist of mosses in India, the typical taxonomic checklists Botanical Survey of India (BSI) and Zoological Survey of India (ZSI) generates. As discussed previously, many potential biases interplay in the generation of these checklists and therefore these are not as valuable as ATBI. Also, note that none of the biological species exists on itself in nature. Species occupy its own position in a complex network of other species in an intricate ecological niche, and therefore the complete picture of overall diversity is far more informative, richer and valuable. For example, it is now universally known that normal microflora, bacteria and viruses inhabiting on the body normally contributes in a number of physiological traits, for example, prevention of diabetes and heart diseases in humans, life history switch and morphology in algae, disease resistance and growth promotion in plants and so on. To generate any form of species inventories, the basic requirement is accurate species identification literature field guide. The most popular taxonomic field guides are the so-called dichotomies identification keys, wherein contrasting and easily identifiable traits. 8. Paper 03 Biodiversity and Conservation. Guide the field researcher from higher taxonomic levels class, order to lower taxonomic levels family, genus and ultimately to the species level identity of a given specimen. These keys are dichotomous, means only two contrasting alternatives of traits are presented, for example, leaf has parallel venation, versus, leaf has reticulate venation. To use such an ID key correctly, the field taxonomist should know all the taxonomic technical terms jargon, which differ substantially across the disciplines. For example, in our earlier example, the identifying person should be competent enough to distinguish various leaf venation systems, and for developing such a technical competency, many years of experience is usually required. Obviously due to the complexity, taxonomic specialists tend to focus on a specific taxonomic lineage, for example marine green algal genus Monostrum or Ulva. Unfortunately, such species identification guides are mostly non-existent in the most speciose habitats of Earth including tropics and aquatic hotspots. This is the main reason for poorer biodiversity documentation in these areas. Newer developments in the species identification arena includes algorithmic approach from field photographs and DNA barcoding. These tools require far lesser expertise, but these have its own shortcomings. Species identification from photographs mostly work in the similar fashion how search by image tool of Google works. It compares the photograph with the database of correctly identified photographs pixel by pixel for the closest match. One such a popular application is Frog Finder, developed by taxonomists from India. It enables anyone with an Android-based smartphone to identify the described frog species by clicking a photo within that app. However, photographs have so many variables affecting the database match decision, for example brightness, color temperature, contrast and so on. It is almost impossible to normalize these field photos for an accurate comparison with that of database. Another problem is morphovariance, morphological variability of the species that depend upon biotic, for example, bacterial flora, or abiotic, for example, seawater salinity factors. This is an issue with any form of morphology-based species. 9. Paper O3 Biodiversity and Conservation. Identification method. Consider Ulva pasquima, a recently discovered species from Indian coast. Intraspecific varieties that grown at low saline estuaries have branched pattern of its thalli, the algal body, while those varieties that grows on the shore have unbranched thalli, and therefore a dichotomous key that call for contrast between branched and unbranched would lead to incorrect identification. Perhaps the only way to alleviate these problems of traditional taxonomic identification is through DNA barcoding, amplification of certain genomic regions, genes, introns, etc., and comparing the DNA sequence with that of the standard database like GenBank for DNA. This DNA barcoding approach is the current gold standard for the species identification, however it has several shortcomings as well. The biggest issue is the cost of DNA sequencing. Although newer methods of next-generation sequencing approaches drastically reduces the expenses involved, the cost is still substantial for taxonomists from developing countries. Another hurdle is that DNA barcoding is impractical for field researchers. It is not possible to sequence DNA molecules from the field, at least as of today. Newer sequencing methods like that of nanopore systems aim to address this issue. The Oxford nanopore sequencer is a very small gadget, like a typical smartphone, and enables rapid DNA sequencing at the field. The technology is rapidly evolving as of this writing, and more developments are expected. Yet another issue with the DNA barcoding is that as it compares the accession in standard repository, the accuracy of this method depends upon the accuracy of matched accession in the repositories. A number of accessions in GenBank database are incorrectly identified and therefore unreliable sources. There is no way to find out which sequences are incorrectly identified among the entries of GenBank, and therefore either manual or automatic curation is impossible. Perhaps a workaround is to rely only on the published sequences, i.e., those accessions generated as part of a published literature, and rely on multiple database accessions rather than one. Barcode of Life database bold is yet another public sequence repository popular among taxonomists. The current standard for species. 10. Paper O3 Biodiversity and Conservation. Identification relies on DNA barcoding and is often combined with at least one of the traditional methods, for example, morphology, life history, or ecology. Specimen vouchers are an indispensable part of any taxonomic study and wider biodiversity documentation. Vouchers are the permanent records of collected specimen accessible in any public repositories, for example museums, herbaria, free storage and so on. In plant taxonomy, vouchers are mostly pressed herbarium sheets of plants showing pertinent morphological features. In earlier days the preferred method for voucher preparation was preservation in formalin, as it prevents microbial degradation and thereby preserves the morphological features. However, formalin preservation invariably degrades the DNA for the subsequent extraction. Many recent DNA barcode-based studies could able to extract and sequence DNA barcodes from more than 3 centuries old holotypes, the voucher specimen that is directly linked with a taxonomic discovery and its new name from herbariums. However, formalin preservation is still popular among zoologists, who, in addition, also preserve tissue materials in liquid nitrogen for long-term storage and subsequent DNA extraction in case deemed necessary in future. Extracted DNA can also be stored for a long time in liquid nitrogen, in the so-called DNA banks. Species distribution maps are essential components of species inventories. The distribution maps overlay species ranges in a geographical map. Print distribution maps, popular earlier, have largely gone out of favor and currently internet-based high-resolution map overlays are universally preferred. These overlays are downloadable files mostly .kml extension compatible with Google Earth, which can be added into Google Earth as a layer to see the distribution map behind the chosen representation of the base geographical map. The map overlays contains known places of species distribution, with accurate coordinates, latitude and longitude data from a GPS device. These map overlays are independently published in the form of floristic and faunal maps of particular regions. Many such maps. 11. Paper O3 Biodiversity and Conservation. Are downloadable from Global Biodiversity Information Facility, GBIF. These maps are invaluable tools for deciding sampling expeditions for field taxonomists and population geneticists, as well as to find whether a taxonomic record is new to a particular area. 3.3. Species characterization. Species characterization encompasses the discovery of new species and other major taxonomic revisions. Therefore, species characterization occupies central part of any biodiversity programs. The current consensus among taxonomists is that the described biodiversity comprise only 10% or fewer of the total biodiversity. Vast majority of the global biodiversity remains to be discovered. This is especially the case with underrepresented taxonomic lineages, underrepresented habitats, and unculturable microorganisms, where almost the entire biodiversity remain undescribed. To discover a new species and to name it, taxonomists need to be a specialist competent enough to comply various codes and articles of nomenclatural systems. These codes differ substantially across disciplines. 3 most widely used codes are International Code of Nomenclature for Plants, Algae and Fungi (ICN), International Commission on Zoological Nomenclature (ICZN), and International Code of Nomenclature of Prokaryotes (ICNP). For viruses 2 systems are used widely. These are the International Committee on Taxonomy of Viruses (ICTV) system and Baltimore Classification System. These codes contain articles detailing how a new species is described, and other rules for various taxonomic revisions, for example, 1 earlier species splitting into 2, 2 known species combining into 1 and so on. In a way these codes resemble the discipline of law and a command over all the articles with several case studies are essential for taxonomic competency essential for any taxonomic revisions. Earlier, ICBN International Code for Botanical Nomenclature insisted that new species descriptions invariably have to be in Latin for preserving the 12. Paper O3 Biodiversity and Conservation. Uniformity. However, as per the latest code ICN, Latin descriptions become optional. Descriptions have to be either in English or in Latin. An article uniform across these codes is what construe effective publication, a publication that can be acceptable for taxonomic revisions and new species descriptions. Generally, a publication available in public repositories, for example, a downloadable ebook in PDF, a published book, an article published in a journal with ICSN number, etc. are all effective publications, while a university dissertation or thesis are not. These codes are very exhaustive and consider all possible controversies. For example, principle of taxonomic priority. The first formal scientific name given to a plant or animal taxon shall be the name that is to be used, called the valid name in zoology and correct name in botany. Once a name has been used, no subsequent publication of that name for another taxon shall be valid zoology or validly published botany. In order to describe a new species, taxonomists should compare the specimen with species descriptions of all other species of the same genus to make sure that the species in question has not yet been described. However, no consensus exists on which morphological features need to be prioritized, or whether a single unique feature could be sufficient to call it a unique species. For example, two life history forms, sexual versus asexual, or two morphological forms, branched or unbranched, among marine algae. As traditional species descriptions are almost entirely based on morphology, intraspecific variants and ecotypes are often described as different species in this system. The only solution for these problems is DNA taxonomy, as described below. 3.4. DNA taxonomy. The term DNA taxonomy refers usage of DNA sequence data for the taxonomic characterization, especially for species discovery and delineation. This term is also used as a synonym for DNA barcoding in many disciplines, however in sensu stricto, DNA. 13. Paper 03 Biodiversity and Conservation. Barcoding refers identification of previously described species using public databases, while DNA taxonomy refers delineation and description of new species. Both these methods make use of identical sequence barcodes, for example, gene coding for Rubisco large subunit in plants RBCL gene, or gene coding for cytochrome C oxidase 1 CO1 gene in animals. These methods first generate the barcode data and perform database searches in DNA barcoding to see whether the newly generated sequences matches with any existing sequences in the database. In that case, species discoveries would be invalid. If no statistically significant match is found, two possible reasons could be either the species in question is previously described, yet no one has generated the DNA barcode data for this species at the selected locus gene or intron, or the species in question is truly new, previously undescribed. To rule out the first possibility, the taxonomist first list out all the species of the genus in question, and generate the missing DNA barcodes, the gaps, non-existent in the database. Paper 03 Biodiversity and Conservation. Barcoding refers identification of previously described species using public databases, while DNA taxonomy refers delineation and description of new species. Both these methods make use of identical sequence barcodes, for example, gene coding for Rubisco large subunit in plants RBCL gene, or gene coding for cytochrome C oxidase 1 CO1 gene in animals. These methods first generate the barcode data and perform database searches in DNA barcoding to see whether the newly generated sequences matches with any existing sequences in the database. In that case, species discoveries would be invalid. If no statistically significant match is found, two possible reasons could be either the species in question is previously described, yet no one has generated the DNA barcode data for this species at the selected locus gene or intron, or the species in question is truly new, previously undescribed. To rule out the first possibility, the taxonomist first list out all the species of the genus in question, and generate the missing DNA barcodes, the gaps, non-existent in the database. If no significant match is found even after this, then it is cogent to conclude that the species in question is truly novel. However, there is no consensus exist among the taxonomists on what construes statistically significant match. A pairwise similarity score of less than 98.7% is generally accepted for eukaryotes. That is, if the nearest match of generated DNA sequence has less than or equal to 98.7%, then this might be a new species. However, this cutoff is lower for prokaryotes, typically 95%. In other words, as per bacterial taxonomy, the entire primates which typically have higher than 98% sequence homology in commonly used sequence barcodes should be called one species. Therefore, bacterial species tend to be more rigorous with lot of intraspecific variants that could be termed individual species as per eukaryotic taxonomy. In the case of fruit fly Drosophila, a single mutation in the genome was enough to erupt a new species. DNA taxonomy relies on phylogenetic inference, wherein a phylogenetic tree, also called phylogram, is generated using a number of methods. Popular methods include maximum likelihood and Bayesian inference. The tree is constructed first by constructing a multiple sequence alignment. To make an alignment, sequence data of suspected new species. 14. Paper 03 Biodiversity and Conservation. Isolates from multiple locations is compared with GenBank using BLASTn, and several of the top hits are downloaded. All these sequences would be subjected to a multiple sequence alignment algorithm followed by a phylogenetic inference program. Detailed descriptions of the same goes far beyond the scope of current module. Finally, in the generated tree, a distinct clade, a cluster encompassing all the generated accessions of species in question bolsters our hypothesis of monopoly, and a new species is per phylogenetic species concept. In strict sense, phylogenetic species concept defines OTIS operational taxonomic units encompassing all descendants of a common ancestor, no descendants as part of other clades, and no non-descendants as part of the clade in question, the so-called reciprocal monopoly. These OTIS might represent a species, or an intraspecific variant, and is so defined to accommodate ambiguities with the term species. 4. Summary. 4.1. It is now estimated that more than 90% of the species on planet Earth remain to be discovered, and therefore taxonomy-based biodiversity characterization is extremely important. 4.2. Amongst the known biodiversity for inventorying, there are several biases at play. Habitat bias, where tropics and aquatic habitats, despite very high level of biodiversity, remain comparatively less described, and lineage bias, where some species-rich lineages like prasinophytes remain poorly described. 4.3. For species inventorying, checklists, all taxa biodiversity information, distribution maps, dichotomous species ID keys and species identification field guides are important. 4.4. Newer and faster methods for species identification involves algorithm-based reverse image lookup in the curated database, as well as DNA barcoding. 4.5. For species characterization, a specialist taxonomist need to extensively follow and adhere with standard codes of nomenclature. 4.6. For species discovery and description, a biphasic approach using one conventional method, often morphology combined with DNA taxonomy, is the current gold standard. 4.7. In the final phylogram, reciprocal monopoly of the generated accessions of species in question is a strong evidence for the OTIS to be erected as a new species. Paper 03 Biodiversity and Conservation. 1. Introduction. Among the 5 major kingdoms of biodiversity, the kingdom Animalia is one of the important one. All the members of this kingdom are multicellular, eukaryotes and heterotrophs. Most of the members can move independently and are regarded as modal. As per estimates, there are around 9 to 10 million species of animals, and among these about 8 lakh species have been identified. India has only 2.4% of global land area but contains 7-8% of globally recorded species. Over 46,000 species of plants and 96,000 species of animals are recorded in India. Animals may lack symmetry or may have radial or bilateral symmetry. Bilaterally symmetrical animals have dorsal and ventral sides, as well as anterior and posterior ends. There are 3 major clades of bilitarian animals: Deuterostomia, Liphotrochozoa, and Ictiozoa. With one exception, the phyla in these clades consist entirely of invertebrates, animals that lack a backbone. Chordata is the only phylum that includes vertebrates, animals with a backbone. They have been classified, subdivided into many categories on different bases as discussed in this module. The module discusses the evolutionary origin of animals, different animal classification systems, animal germ layers and their derivatives, state animals and birds, etc. 2. Evolutionary origin of animals. The history of animals spans more than 500 million years. Fossil biochemical evidence and molecular clock analyses indicate that animals arose over 700 million years ago. Genomic analyses suggest that key steps in the origin of animals involved new ways of using proteins that were encoded by genes found in choanoflagellates. This table provides an overview of how animals evolved from their distant common ancestor over 4 geologic eras. Era period age millions of years ago. Important events. Neoproterozoic Ediacaran 560 to 542 soft-bodied, radially symmetric invertebrate animals appear fossils. Mossonite spriggi. Sprigina flounder C. Cloudina. Paper 03 Biodiversity and Conservation. Dickinsonii costata. Paleozoic Cambrian 542 to 488 sudden increase in diversity of many animal phyla Cambrian explosion. Fossils. Pichia eel-like chordate. Morella small arthropod. Anomalocaris large animal with grasping limbs and a circular mouth. And Hallucigenia animals with toothpick-like spikes. Ordovician 488 to 444 colonization of land by animals. Invertebrates dominated Silurian 444 to 416 radiation of crinoids, a continued proliferation and expansion of the brachiopods, and the oldest known fossils of coral reefs, also marks the wide and rapid spread of jawless fish. Devonian 416 to 359 diversification of bony fishes, first tetrapods and insects appear fossils. Coccostiuscuspidatus, Tiktaalik. Carboniferous. 359 to 299 origin of reptiles, amphibians dominant. Permian 299 to 251 radiation of reptiles, origin of most present-day groups of insects, extinction of many marine and terrestrial organisms. At end of period fossils, Dimetrodon. The largest known carnivore of its day. Paper 03 Biodiversity and Conservation. Mesozoic Triassic 251-199.6 dinosaurs evolve and radiate, origin of mammals Jurassic 199.6-145.5 dinosaurs abundant and diverse fossil. Romaleosaurus victor, a plesiosaur Cretaceous 145.5-65.5 dinosaurs become extinct at end of period Cenozoic Paleogene 65.5-23 major radiation of mammals, birds, and pollinating insects, origins of many primate groups. Neogene 23-2.6 continued radiation of mammals. Appearance of bipedal human ancestors Quaternary 2.6-0.01 Ice Ages, origin of genus Homo. 3. Animal classification. 3.1. Carolus Linnaeus classification. All modern classification systems have their roots in the Carolus Linnaeus classification system. His two most important contributions to taxonomy were a hierarchical classification system. Species are grouped in increasingly broad taxa. Related genera are placed in the same family. Families in orders, orders in classes, classes in phyla, phyla in kingdoms. The system of binomial nomenclature, a two-part naming method. Genus, the first part of the binomial to which the species belongs. The first letter of the genus is always capitalized. Specific epithet. The second part of binomial is unique for each species within the genus. Entire binomial is italicized and scientific names are in Latin. 3.2. Five kingdom classification. Many biologists including R. H. Whitaker recognized five kingdoms. Monera prokaryotes protista, a diverse kingdom consisting mostly of unicellular organisms plantae, fungi, and animalia. However, phylogenies based on genetic data soon began to reveal a problem. With this system, some prokaryotes differ as much from each other as they do from eukaryotes. Such difficulty led biologists to adopt a three-domain system. 3.3. Three-domain classification. Tree of life. The three-domain system is a biological classification introduced by Carl Woese et al. In 1977 that divides cellular life forms into archaea, bacteria, and eukaryote domains. Tree of life is based in part on sequence comparisons of rRNA genes or rDNA. Tree of life is based in part on sequence comparisons of rRNA genes. 3.4. Other animal classification systems. Paper 03 Biodiversity and Conservation. Paper 03 Biodiversity and Conservation. Animal classification based on level of organization. At cellular level of organization, animal kingdom is divided into two subkingdoms, protozoa and metazoa. Protozoa are defined as single-celled eukaryotic organisms and metazoa are multicellular eukaryotic organisms. The subkingdom metazoa is further subdivided into two branches, parazoa and eumetazoa. Parazoa. Cells are arranged as loose cell aggregates, i.e., they exhibit cellular level of organization, absence of tissue or organ system level of organization. E.g., phylum Porifera. Eumetazoa. Clade comprising all major animal phyla except Porifera. Embryo gets layered during gastrulation. Exhibit either tissue or organ system level of organization. B. Tissue level of organization. In phylum Cnidaria and Ctenophora, the arrangement of cells is more complex. Here the cells performing the same function are arranged into tissues. C. Organ level of organization. Organ level is exhibited by members of platyhelminths and other higher phyla where tissues are grouped together to form organs, each specialized for a particular function. E. Animal classification based on symmetry. At symmetry, animals can be categorized on the basis of their symmetry. B. Asymmetry. Sponges are mostly asymmetrical, i.e., any plane that passes through the center does not divide them into equal halves. C. Radial symmetry. When any plane passing through the central axis of the body divides the organism into two identical halves, it is called radial symmetry. Cnidarians, ctenophores and echinoderms have this kind of body plan. In phylum Echinodermata, larval stages are bilaterally symmetrical and adult stages are radially symmetric. Paper 03 Biodiversity and Conservation. D. Bilateral symmetry. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. E. Animal classification based on number of germ layers. In all the animals except sponges, the embryo becomes layered during gastrulation. As development progresses, these layers, called germ layers, form the various tissues and organs of the body. At diploblastic animals, animals in which the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals, e.g., cnidarians and ctenophores. An indifferentiated layer, mesoglia or mesoel, is present in between the ectoderm and the endoderm. B. Triploblastic animals. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm, are called triploblastic animals, platyhelminths to chordates. IB. Animal classification based on body cavity. A body cavity, presence or absence of a cavity between the body wall and the gut wall, is very important in classification. B. Coelomates. Most triploblastic animals have a body cavity, a fluid or air-filled space located between the digestive tract and the outer body wall. This body cavity is also called a coelom. Animals possessing coelom are called coelomates, e.g., annelids, mollusks, arthropods, echinoderms, and chordates. C. Pseudocoelomates. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom and the animals possessing them are called pseudocoelomates, e.g., aschelminths. D. Acoelomates. The animals in which the body cavity is absent are called acoelomates, e.g., platyhelminths. Paper 03 Biodiversity and Conservation. B. Animal classification based on presence or absence of notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Non-chordates. Those animals which do not form notochord structure are called nonchordates, e.g., porifera to echinoderms. B. Chordates. 4 key characters of chordates: a notochord, a dorsal, hollow nerve cord, pharyngeal slits or clefts, and a muscular, postanal tail. B. Animal classification based on embryonic development and segmentation. Embryonic development. Based on certain aspects of early development, many animals can be described as having one of two developmental modes: protostome development or deuterostome development. These modes can generally be distinguished by differences in cleavage, coelom formation, and fate of the blastopore. B. Protozomes. Protostome development begins with spiral, determinate cleavage. The coelom forms from splits in the mesoderm and the mouth forms from the blastopore. Protozomes include the phyla Mollusca, Annelida and Arthropoda. C. Deuterostomes. Deuterostome development is characterized by radial, indeterminate cleavage. Coelom forms from mesodermal outpocketings of the archenteron and the mouth forms from a secondary. Anus develops from blastopore. Deuterostomes include the phyla Echinodermata and Chordata. Paper 03 Biodiversity and Conservation. 4. Description of animal. 4. Description of animal phyla. Phylum description. Porifera sponges lack true tissues. Choanocytes ingest bacteria and tiny food particles. Body is supported by a skeleton made up of spicules or sponging fibers. Sexes are not separate hermaphrodite. Paper 03 Biodiversity and Conservation. Reproduce asexually by fragmentation and sexually by formation of gametes. Examples: Cycon, Cypha, Spongula, freshwater sponge, and Euspongia, bath sponge. Ctenophora, comb jellies. 8 rows of ciliated combs, or cetens. Bioluminescence is well marked in ctenophores. Reproduction takes place only by sexual means. Examples: Pleurobrachion, Ctenoplana, Cnidaria, unique stinging structures, nematocysts housed in specialized cells. Cnidocytes. Corals have a skeleton composed of calcium carbonate. Cnidarians exhibit 2 basic body forms called polyp and medusa. Those cnidarians which exist in both forms exhibit alternation ageneration, metagenesis, e.g., Obelia. Examples: Physalia, Portuguese man of war, Adamsia, C. anemone, Penatula, C. pen, Gorgonia, C. fan, and Meandrina, brain coral. Platyhelminths, flatworms. A central nervous system that processes information from sensory structures. Specialized cells called flame cells help in osmoregulation and excretion. Development is through many larval stages. Some members like Planari possess high regeneration capacity. Examples: Taenia, tapeworm, fasciola, liver fluke. Paper 03 Biodiversity and Conservation. Nematoda. Roundworms, cylindrical pseudocoelemates with tapered ends. Undergo ecdysis. Alimentary canal is complete with a well-developed muscular pharynx. An excretory tube removes body wastes from the body cavity through the excretory pore. Examples: Ascaris roundworm, Wetteraria, filaria worm, Ancylostoma hookworm. Annelida, segmented worms. Segmented body wall, metameres, and internal organs, except digestive tract, which is unsegmented. Aquatic annelids like Nereis possess lateral appendages, parapodia, which help in swimming. Nephridia, sing. Nephridium help in osmoregulation and excretion. Reproduction is sexual. Examples: Nereis, Ferritima, earthworm, and Hirudinaria, blood-sucking leech. Mollusca, second largest animal phylum. Body parts include muscular foot, visceral mass, mantle. Coelom reduced. Most have hard shell made of calcium carbonate. Mantle cavity in which feather-like gills are present. The mouth contains a file-like rasping organ for feeding, called radula. Examples: Pila, apple snail, pinctada, pearl oyster, sepia, cuttlefish, loligope, squid, octopus, devilfish, aplysia, sea hare, dentalium, tusk. Paper 03 Biodiversity and Conservation. Shell and Chaetopelura, chitin. Arthropoda, largest phylum of Animalia. Segmented body, jointed appendages, an exoskeleton made of protein and chitin. The body consists of head, thorax, and abdomen. Respiratory organs are gills, book lungs, or tracheal system. Circulatory system is of open type hemolymph. Sensory organs like antennae, eyes, compound, and simple statocysts or balance organs are present. Excretion takes place through Malpighian tubules. Examples: economically important insects: apis, honeybee, bombyx, silkworm, laxifer, lac insect vectors, Anopheles, Chilixantha aedes, mosquitoes, gregarious pest, locust living fossil, limulus, king crab. Echinodermata, echinoderms. The adult echinoderms are radially symmetrical but larvae are bilaterally symmetrical. The most distinctive feature of echinoderms is the presence of water vascular system which helps in locomotion, capture and transport of food and respiration. An excretory system is absent. Examples: Asterias, starfish, echinus, sea urchin, antodon, sea lily, cucumaria, sea cucumber, and Ophiora brittlestar. Chordata, chordates characterized by the presence of a notochord, a dorsal hollow nerve cord, and paired pharyngeal gill slits. Paper 03 Biodiversity and Conservation. Urochordata and cephalochordata. Subphyla Urochordata and cephalochordata are often referred to as protochordates. In Urochordata, notochord is present only in larval tail, while in cephalochordata, it extends from head to tail region and is persistent throughout their life. Examples: Urochordata, Acidia, Salpa, Doliolum, Cephalochordata, Branchiostoma, Amphioxus, or Lancelet. Vertebrata. The members of subphylum Vertebrata possess notochord during the embryonic period. The notochord is replaced by a cartilaginous or bony vertebral column in the adult. Vertebrates have a ventral muscular heart with 2, 3 or 4 chambers. Kidneys for excretion and osmoregulation and paired appendages which may be fins or limbs. Class Cyclostomata. Elongated body bearing 6 to 15 pairs of gill slits for respiration. Cyclostomes have a sucking and circular mouth without jaws. Examples: Petromezone lamprey and Myxine hagfish. Pisces fish. Class Chondrichthyes. Paper 03 Biodiversity and Conservation. Notochord is persistent throughout life. Gill slits are separate and without operculum, gill cover. The skin is tough, containing minute placoid scales. Due to the absence of air bladder, they have to swim constantly to avoid sinking. Heart is 2-chambered, 1 auricle and 1 ventricle. Some of them have electric organs, e.g., torpedo, and some possess poison sting, e.g., trigon. They are cold-blooded poikilothermous animals, i.e., they lack the capacity to regulate their body temperature. Many of them are viviparous. Examples: Scoliodon, dogfish, Christie's, sawfish, carcharodon, great white shark, trigon, stingray. Class Osteichthyes. It includes both marine and freshwater fishes with bony endoskeleton. They have 4 pairs of gills which are covered by an operculum on each side. Air bladder is present which regulates buoyancy. They are mostly oviparous. Examples: marine, Exocetus flying fish, hippocampus, seahorse, freshwater, labio, rohu, catla, catla, clarias, magur, aquarium, betta fighting fish, pterophyllum, angelfish. Tetrapoda. Paper 03 Biodiversity and Conservation. Class Amphibian. Body is divisible into head and trunk. The amphibian skin is moist without scales. The eyes have eyelids. A tympanum represents the ear. Alimentary canal, urinary and reproductive tracts open into a common chamber called cloaca which opens to the exterior. Respiration is by gills, lungs and through skin. The heart is 3-chambered, 2 auricles and 1 ventricle. These are cold-blooded animals. They are oviparous. Examples: bufo, toad, rana, frog, hyla, tree frog, salamandra, salamander, Ichthyophis, limbless amphibia. Class Reptilia. Body is covered by dry and cornified skin, epidermal scales or scutes. They do not have external ear openings. Tympanum represents ear. Heart is usually 3-chambered, but 4-chambered in crocodiles. Reptiles are poikilotherms. Snakes and lizards shed their scales as skin cast. They are oviparous. Examples: chelone, turtle, testudo, tortoise, chameleon, tree lizard, calotes, garden lizard, crocodilus, crocodile, alligator, alligator, hemidactylus, wall lizard, poisonous snakes, naya cobra. Paper 03 Biodiversity and Conservation. Bongarus, crite, vipera, viper. Class Aves. Presence of feathers and most of them can fly except flightless birds, e.g., ostrich. The forelimbs are modified into wings. The hind limbs generally have scales and are modified for walking, swimming or clasping the tree branches. Skin is dry without glands except the oil gland at the base of the tail. Endoskeleton is fully ossified, bony, and the long bones are hollow with air cavities, pneumatic. The digestive tract of birds has additional chambers, the crop and gizzard. Heart is completely 4-chambered. They are warm-blooded homoothermous animals, i.e., they are able to maintain a constant body temperature. They are oviparous. Examples: corvus, crow, columba, pigeon, psittacula, parrot, struthio, ostrich, pavo, peacock, aptenidites, penguin, neophron, vulture. Class Mammalia. The most unique mammalian characteristic is the presence of milk-producing glands, mammary glands, by which the young ones are nourished. The skin of mammals is unique in possessing hair. External ears or pinnae are present. Paper 03 Biodiversity and Conservation. Different types of teeth are present in the jaw. Heart is 4-chambered. They are homeothermous. They are viviparous. Examples: oviparous ornithorhynchus, platypus, viviparous, macropus, kangaroo, pteropus, flying fox, camelus, camel, macaca, monkey, ratus, rat, canis, dog, felis, cat, elephas, elephant, equus, horse, delphinus, common dolphin, balinoptera, blue whale, pantheratigrus, tiger, pantherleo, lion. Paper 03 Biodiversity and Conservation. 5. Salient features of different phyla in the animal kingdom. Phylum level of Organization. Cymot Rye. Body cavity segmentation. Digestive, circulatory, respirator Y, nervous system. No. Of species. World E. 6. Animal germ layers and their derivatives. Triploblastic animals. Germ layer body tissues derived. Ectoderm, epidermis. Epidermis and epidermal derivatives: hair, nails, tooth enamel, mammary, sweat and sebaceous glands, epithelia of skin, ear, eye and nose, lower anal canal, cornea and lens of eye, epithelium of anterior pituitary, sensory receptors. Neural tube. Central nervous system, retina, neurohypophysis, pineal gland, astrocytes, oligodendrocytes. Neural crest. Connective tissue and bones of the face and skull, cranial nerve ganglia, C cells of thyroid gland. Paper 03 Biodiversity and Conservation. Conotruncal septum in the heart, odontoblasts, dermis in face and neck, spinal dorsal root ganglia, sympathetic chain and preortic ganglia, parasympathetic ganglia of the GI tract, adrenal medulla, Schwann cells, glial cells, meninges, forebrained melanocytes. Mesoderm, paraxial mesoderm or somatic mesoderm. The sclerotome forms cartilage, the syndotome forms tendons, the myotome forms skeletal muscle, the dermatome forms the dermis as well as skeletal muscle, and endothelial cells. The head mesoderm include connective tissue and the muscles of the face. Intermediate mesoderm. It develops into the part of the urogenital system, kidneys and gonads, as well as the reproductive system. Lateral mesoderm. The lateral plate mesoderm splits into parietal, somatic, and visceral splanchnic layers. The visceral layer forms the wall